All right, so we are going to talk today about transport system across the cells, across the plasma membrane. Right, so we have covered already the structures of the plasma membrane, so keep that in mind while we talk about that. So first we'll talk about the passive transport system, diffusion, and then we'll get into active transport. What is passive transport system? Passive transport system, like the name is telling you, it's passive. There is no energy required for passive transport system. The molecules just go like diffuse across their concentration gradient. So it will diffuse from where it is present in low concentration, then it will go in air wherever it is in present in high concentration, and then it will diffuse in the area of low concentration, and it will try to make everything in equilibrium. So wherever it is present more, it will try to diffuse in the area where it not so much so present. Okay, so and they will try to make an equilibrium. Difference between active and passive. So like I said, passive does not need any energy from outside, but active might need it. So, structure of plasma membrane, does it help in diffusion? Yes, it does. First of all, the ways molecules actually transport in and out in the cell or out the cell, that is through plasma membrane, right? So when this happens, then there are two mechanisms that are like working in the background. First is the active transport, where ATP, which is our energy molecule, that is required, and in the passive transport, when this ATP is not required. All right, so that is a passive transport. But that does not mean that plasma membrane will allow everything to come inside the cell. It will only allow certain substances to cross and come inside the cell. Doesn't matter what pathway it is following, active or passive, but only certain molecules. And that's why plasma membrane is called as selectively permeable. Okay. So, how does this work? So, firstly, we'll talk about the passive membrane transport. First, diffusion. Okay, diffusion simply means it is diffusing from one place to another place. Okay, it can be simple, it can be facilitated. Means simple means it's simply diffusing. Facilitating means something is facilitating it. Then there is a special type, osmosis and channel mediated. Now, all of these diffusion mechanisms are influenced by three factors: concentration of the molecule, molecular size, heat or temperature. So you have learned that in the in chapter 2 that if you apply a little bit heat to a chemical reaction, then it's always the reaction goes faster, same as here. So if something is diffusing, you are applying a little bit more temperature, it will diffusion rate will be more. So more is the temperature, that will be directly proportional to diffusion, the diffusion rate. Okay, more the temperature, more is the diffusion. If the molecular size is bigger, okay, big in size, that will take comparatively larger time to diffuse. But if it is small, it can run around pretty easily. So molecular size, if the molecular size is higher, then that means the diffusion rate will be slower. Okay, now what about concentration? So concentration, more the difference is the concentration, because we just discussed that how substance can diffuse from high to low concentration. So more the difference is in concentration present in a into solutions, or in among the in that particular in one situation, suppose, more will be the diffusion rate. For suppose you have more the difference in concentration, that will means more in diffusion rate. Okay, difference in concentration. So suppose you have one here where there is lots of these molecules present, and you connect it with another tube where like very few of is present. So diffusion rate will be really high, because it will the molecules will move from this one to this one, from A to B, suppose, and then the diffusion rate will be really higher. All right, now here is an example. Suppose you have a dye pellet in a solution, suppose it's water. So first of all, the dye is super highly concentrated around this area. Okay, so this is highly concentrated, and then here on the top of the water, suppose it's low, it's not that much dye present, so it will start diffusing from high to low area, and it will continue to do so until all everything is equally distributed across the water. So same principle is applied in cell membrane also. So suppose you have a lipid, any solute or anything that is present more. For example, here in this picture, the lipid soluble solutes present more outside and outside the cell. This is extracellular fluid, and but less in the cytoplasm in the cell. It will diffuse really faster. It will try to establish equilibrium, and that's why this is called as simple diffusion. Okay, a substance like a lipid soluble substances which is small enough, they can and also can pass through the lipid structure of phospholipid. They will, because these are lipid soluble, so they can easily will be able to pass through. Therefore they will easily just pass across the membrane and will go from high to low concentration. That's simple diffusion. What about carrier mediated? The principle is still the same. They will all diffuse from area of high to low, but here this is a carrier mediated. So carrier means this is the carrier is the protein that's sitting on the membrane, right? They will enable certain substances to get inside. Okay, for example, certain lipid insoluble solutes like sugars or amino acids, so they are in lipid insoluble, so it won't be very easy for them to pass through, because plasma membrane have lipid in their in their cell membrane, right, in the cell membrane structure. So then they will take help of this carrier, so this carrier will actually enable them to pass through the membrane, but again diffusing from high to low concentration. And these carriers are nothing but proteins that's on the membrane, protein carriers. Similarly, channels, and when we're talking about channels, it's simply these protein channels. All right, again, certain lipid soluble solutes that cannot pass through directly through the plasma membrane, they will take like a pathway. They might take a channel mediated pathway, and this channel is a protein channel. What about in case of osmosis? Osmosis is a special type of diffusion that typically can happen with water. So in this case also, water will also go through the high to low concentration, but it might take a special kind of carrier called as aquaporine. Aquaporine only passes water, so water will pass through that, all right, with aquaporine. But the other hand, the water molecule actually can pass through plasma membrane directly as well, so it can take both pathways. And this is called as the osmosis, diffusion of a solvent like water through a specific channel protein like aquaporine. So what is osmolarity, if I have to, because we just spoke about osmosis. So what is osmolarity? Is the measure the concentration of the total solute particles in a solvent. Suppose how much sugar is present in water, 5 grams, 6 grams, 10 grams, that's in maybe 100 ml water, 200 ml water, that is osmolarity. Now water concentration actually varies with solute particles, because solute particles can displace water. Suppose you have water and you only have like 5 grams of sugar into it. Okay, now if you keep the water same, but if you add 15 grams of water, suppose, so naturally this will displace the water because there is more water in there. So that's what it is telling you. So more, if you put solute, that the water concentration will go down, right? And but water moves from areas of low solute to high area solute, or simply high water to low water concentration. So this is diffusion of water through aquaporine, because diffusion of water across the selectively permeable membrane, when it follows this path, it actually follows the hydrostatic pressure. I mean the pressure difference that is created by water, and what is the different between the outside of the cell and inside of the cell. So more the hydrostatic pressure is, more will be the water flow. To simplify it, basically when water diffuses through these aquaporine channels in plasma membrane from high to low concentration, that's when we will call it as osmosis of water. Okay, and these things can lead up to hemolysis and crenation, which we will talk about in a second. So let's go through this example of osmosis. Like this similar, imagine the same thing can happen in cell membrane. Suppose this is a permeable membrane, okay, and you have this tube. On this side of the tube, suppose side A, and this is side B, you have less solute present and more solute is present in side B. Okay, so what will happen? So therefore the solute will try to establish equilibrium. So here it is present in high concentration, high solute, and here it is present in low in concentration, so low solute, right? So therefore, since it will try to establish equilibrium, it will try from B, it will get transferred from B to A, because high to low, B to A, right? On the other hand, therefore, if solute is moving in this direction, water will go in that opposite direction, because water is present, solute is less here, therefore water is more here, in this way, in this tube, more water. And here we have less water. Why? Because here is more solute, and inside B, right? Because we did just discuss that if we add more solute in a water, then that will displace the water, right? So here we have more solute present, so we have less water inside B. Here we have low number of solute present, so we have more water. And remember, the level is still the same, so it has to be the water has been displaced. So high solute, less water inside B, low solute, more water inside A. Now the everything will move from high to low. Therefore, solute will move from B to A, but the water will go in exactly reverse direction, A to B, right? And after a point, they will be in equilibrium. Okay, so this is what we're talking about when we're talking about osmosis happening through a semi-permeable or a permeable membrane. All right. Now we can apply this in the concept of cell. So suppose we take a size of RBC, okay, or we take a cell RBC. So RBC itself has NaCl, sodium chloride, present in cells, right? Or any cell will have. So normal concentration of NaCl in human cells is 0.9%. Okay, in any cell, NaCl concentration, NaCl is present in 0.9% concentration. That's normal. Now that's true in here as well, in RBC. So RBC have 0.9% of NaCl. So suppose there is NaCl 0.9%, and you're putting in an isotonic, iso means same, you're putting this into a solution where there is also 0.9% NaCl. So it's all the same, so no movement will happen here. So this kind of situation, we call it as isotonic solution. The cells retain their normal size and shape, nothing changes. But if you put the cell in a concentration where, suppose this has 0.9%, but NaCl, now that's a normal, but if you put it in a solution that has 2% NaCl, right? So this is an hypertonic solution. So since here is more NaCl present, so this is, we're talking about NaCl here. Okay, since here is more NaCl present, so there is less water here, right? Less water. But since there is less amount of NaCl here compared to outside, so here we have more water in the cell normally. So what would happen in this case? Water will move from high to low. So water will move from high to low concentration, and more sodium chloride will start moving inside, because again going from high to low. In this process, the cell will start, so it's just losing water. Like you can see here, it's losing the water. Okay. They lose water, and they will actually, after losing water, it will look like shrinked, like the cell is looking. So after you have put it in a 2% solution, so therefore if you put cell in a hypertonic solution, cell will shrink. Actually there will be osmosis, water osmosis, and it will shrink. All right, so this is, again, you are putting the cell itself as 0.9% NaCl, and you're putting in a hyper concentration, which is suppose 2%. It can be anything, just I give you an example. Exactly the opposite, you have 0.9%, and now you're putting it, suppose, in a 0.1% solution. Okay, let's draw it out here. So suppose this is 0.9% NaCl here, and you're putting it in 0.1%. Exactly opposite thing will happen. So water will actually start moving in, because there is more water here, right? So water will start moving in because there is less solute outside, so therefore more water, and there is more solute inside, so there is less water, so water is high outside, water will start moving inside, and then the cell will actually have more water than it can accommodate, so then the cell will burst. Okay, it will bloat and then it will burst or lyse. Okay, these, remember this concentration. The bottom line of the story that you have to remember, wherever there is more solute, there is less water, or vice versa. All right, so what is the energy source of all types of diffusion? Well, energy source, will come to that. Let's first talk about number 4. What determines the direction of any diffusion process? It would be the difference in concentration that is present, and right, difference in concentration. What is the energy source of any diffusion? Suppose, let's because diffusion is a passive movement. So how did it happen? There is no energy molecule that is helping it. Like in active there will be ATP, but here there will be no. But the energy source is basically the kinetic energy that is inside these molecules itself. They will help the molecule to move. So the kinetic energy in every molecule, suppose whichever one you're talking about, water, let's say water, or let's say any of these lipid insoluble soluble molecules that we spoke about, they have their own kinetic energy, right? So they will use their own energy to move around, provided there is a concentration difference, otherwise they will not. Okay, so the first is basically the kinetic energy, and what determines the direction is the concentration difference. All right, the next thing we'll talk about is the active transport system. Okay, so active transport system, so we will actually try to differentiate between the active primary and secondary active transport. We'll talk about some special type of transport that happens like in and out of cell, exocytosis, endocytosis, and then membrane potential, and finally CAMs. So active transport simply means it requires energy, right? So suppose, and it typically happens through sodium potassium pump. So we have sodium potassium pump present on the cell membrane, and what they do, they either pull in or out sodium and potassium. Okay, only dedicated for sodium potassium. So either they will put potassium out, mostly they put potassium inside the cell and sodium outside the cell. Okay, potassium is always present more inside the cell and sodium is always present more outside the cell, and that is done, this condition is established by the sodium potassium pump. And who drives this? Obviously ATP, because this is active transport. So this is primary active transport. What is secondary active transport? Suppose sodium potassium pump is already doing this kind of pumping, like it is helping sodium to move inside or outside. So it's kind of in a working condition, and it is mostly when the sodium potassium pump is putting sodium inside. Along with sodium, sometimes glucose also can come, as with the help of these transporter proteins, glucose also can come inside. So the primary concept here is to transport sodium inside, but along with that, glucose also can tag along through these channel proteins. Okay, as this is like through this transporter protein. So glucose is co-transported here. So that is called as secondary active transport. This process also requires ATP. Now vesicular transport, this is another form. So not only through plasma membrane actually cell can transport, but they also form tiny things called vesicles. So what is and the first type of vesicular transport is called endocytosis, means something is moving inside the cell, whatever it is, but it is moving inside the cell. 3 types of it: pinocytosis, phagocytosis, receptor mediated. Pinocytosis means, suppose these are the substance that has to move in, so it will gather around and it will form a vesicle like this. After a point, this vesicle will pinch off from the cell membrane, and whatever needs to be transported inside will be inside the vesicle and it will be in the cell. So this vesicle kind of works like a transport system, or like a car or a bus, train. So whatever has to be done, it will be accumulated within the vesicle. The vesicle will pinch off and then it will like get inside. Similar situation happens in phagocytosis. Only thing, it forms a structure called pseudopod, and this is typically applied for bigger substances. So it's sort of, suppose a huge substance is there, the cell will form, like engulf it around, and it will form a structure called pseudopod, and then finally this will pinch off and this big larger substances will get inside like that. There's receptor mediated. For example, certain substances, maybe they're not that general, so they need to associate with a receptor. So suppose these are those special type of substances, like some special proteins, special ions. So they will have their specific receptor on the plasma membrane. So they will sit on these receptors, and then there will be a vesicle will be formed, and similar to what happened before, the vesicle will pinch off. Now the substances are already associated with their receptors, so the whole thing will be transported inside. So pinocytosis is the most simpler one. Phagocytosis is for larger substances, and therefore it forms pseudopods. And receptor mediated means there is a receptor involved. For specific type of substances will actually get transported like this. Exactly the opposite is exocytosis, where cell actually put, like, throw things out. So it basically forms the vesicles, but then whatever is in the vesicle, that vesicle is thrown out. So the way substances move out of cell, that is called as exocytosis. This is a microscopic photograph of exocytosis. Now, what is membrane potential? We are going to discuss a lot of this later in the semester, but one thing you need to, the basic concept that you need to understand here, that ion concentration is not the same inside the cell, outside the cell. Mostly, like I said, potassium is always present more in the cell, and sodium is present more outside the cell. But, and this definitely creates a charge difference in the cell membrane rather, and that is called as the membrane potential. It's basically the charge or the charge difference. Okay, the charge difference, or the because these ions have their own charge, so moving them around creates a charge difference on the cell. Okay, that is called as the membrane potential. Membrane typically is in resting membrane potential, that it maintains throughout in normal condition, but that can change if these ions start moving around. We will discuss a lot of them later, and potassium is a key player in this transport system. Okay, and this actually is an active transport method. Now this is the cell movement. Now, how cell keeps contact with each other? Firstly, they form CAM addition molecules, which are nothing like, we know we have those glycoprotein complexes or on glycocalyx. So basically, every cell has thousands of these small glycoprotein that forms. So here is the membrane, and here are the glycoproteins. So these glycoproteins are the sticky substances, they actually anchor with each other. So one cell's glycoprotein will, like, combine with the other cell's glycoprotein, and they're sticky, so they will stick around each other. Okay, for example, WBC does this all the time, particularly when they're trying to attack a foreign substance. Another thing that we'll talk about is the plasma membrane receptor. This is also another way a cell actually keeps in contact with each other. How do they do it? Basically, they have specific receptors. We just now spoke about these receptors, right? So cell will have receptor for different kind of substances on the cell membrane. So and these receptors either can do contact signaling, means involve touch between the receptor and then certain substances. So this is an example of that kind of contact signaling. It is making contact with this is the receptor and this is the substance, although this relationship is very specific. And then chemical signaling means involve the binding of a chemical signal to the membrane receptor, and then that basically specific chemical substance will bind to the receptor. That's called chemical signaling. But overall, receptors are on present and they have their own substances. So receptors will recognize those substances, and those substances will be brought into the cell. So the way cell then maintains connection with each other, either through CAMs, glycoproteins, or through these receptors. All right. So I will conclude this lecture right now. Please listen to this before you come on Monday. Some of the concepts we have already discussed in class, and some we will repeat again on Monday. All right. Keep working on the concept checks, and see you on Monday.